I would like to present to you our offerings in advancing gene cell therapy using iPSC and gene editing technologies. So our company are established based on two core technologies. One is iPSC technologies, and the other is gene editing technologies. For gene editing, we have two complementary technologies. One is CRISPR-Cas9 technology, and the other is TARGATT gene knocking technology. So we use iPS cells for cell-based assays, and we also generate patient-specific iPS cells for disease models. And using gene editing, we can generate gene-edited cell line models as well as animal models. And we also use it for bioproduction, particularly in CHO cell, gene modification for bioproduction, and HEC293 cells for antibody screening. And in this particular talk, I would like to focus on our capability of using these technologies to support gene and cell therapy products development. So we're very serious about our quality. So our company, our facilities are ISO certified, ISO 9001 and ISO 13485. And our animal facilities are AAA LAC certified. And we have over 1,000 customers worldwide. And a lot of those are major biotech and pharmaceutical clients. And so this just shows our umbrella quality management systems that monitors all aspects of ASC operations and functions, including CAPA and record management, documentation, regulatory compliance, and all these details. And so our lab are biosafety level 2, and we operate and we perform all the studies in a manner that are consistent with principles of GLP. We also take our cybersecurity very serious in our company, and our data are backed up daily onto cloud as part of our disaster recovery plan, and all access to applied stem cells data and information is controlled by password protected accounts. So, so what are the main focus areas in gene cell therapy? So for gene and cell therapy, there are 3 main phases: preclinical development, clinical, and then, of course, very importantly, CMC, manufacturing. And for preclinical, there are preclinical point of approval concept of experiments, usually using cell or animal models to study efficacy and pilot toxicology. And then for clinical, of course, there are a lot of sample testing that involves assay development, validation, and testing. And for manufacturing, CMC, a lot of testing needs to rely on analytic methods development for drug release, such as potency assays, and also validation, product validation, and assay development. So as I mentioned at the beginning, so ASC's core technologies are iPS cell technology and gene editing. So using those technologies, we're well positioned to generate both cell and animal models, genetically modified cell and animal models. And using iPS cells, we can utilize basically either patient-specific iPS cells or health iPS cells and make into patient cells. And this provides all of the virtually all cell types in the human body, because iPS cells can be differentiated into all kinds of lineage cells. So these cells can be used to develop cell-based assays. So here are the current offerings that Applied Stem Cell can offer to support gene therapy, preclinical, and assay development. So we can provide product vector construct optimization. So for example, for AAV gene therapy, we are experienced with optimizing the expression vector, including promoters, insulators, enhancers, and poly-sequence or codon optimization of the therapeutic gene. And we also experienced with various AAV serotypes and perform infectivity in different cell types as well as in animal models. And we can measure mRNA and protein level expressions. And we are also able to do proof of concept in vivo functional efficacy studies. These are usually done in mouse, in animal models. And we are able to do in vivo biodistribution and viral shedding in mouse models. And so we use qPCR to detect viral DNA copy numbers in various tissues and fluids. We perform dose range studies in mice for pilot toxicology study. And we also develop potency assay for specific product. So this is usually based on using particular cell line and measured either at the DNA or RNA or protein levels. So in the next few slides, I will use some of the data that we have generated developing a Hemophilia A gene therapy product. So this is a gene editing product for Hemophilia A. So Hemophilia A is caused by missing factor A expression in humans. So in this particular therapeutic product, we're trying to express the human factor A, basically restore the expression of human factor A in human liver. So these are some of the preclinical studies that we carried out to show the pilot efficacy and toxicology and the construct optimization studies. So this first series of studies I'm showing here is optimizing gene editing construct. So this is a gene editing product. So we were trying to first look at what kind of construct will be best in terms of efficiency in knock-in, because basically this is a site-specific knock-in using CRISPR. And to do that, we tested 3 different constructs in this example by varying different sizes of the homologous arms, the 5' arm and 3' arms. So the top construct is a long homologous arm, and the middle one is a short arm, and the last one, the lower one, has no homologous arms. Basically, it has a PAM sequence that can be recognized by guide RNA. And then on the right are the FACS data to show the efficiency of gene knock-in. So the higher the number is, the better the efficiency is. So as you can see, the lowest construct gives the best knock-in efficiency, which is 6%, compared to the middle one, which is about 1%, and the top one is about 1.5%. So through this experiment, we decided that the lower construct is the best construct to use for this particular product development. And so then the next thing we want to do is to do in vivo proof of concept functional study. But before we can do that, we have to generate a hemophilia A mouse model. So as I mentioned, hemophilia A is caused by factor A deletion. So we created a model that basically we're knocking out the mouse factor A gene. So we used the CRISPR-Cas9 method to delete starting from exon 4, and then basically causes a frame shift at the end. So there's no factor A gene expressed in this mouse model. So we verified by genotyping here, and then we also verified by functional studies. So this panel C shows that the knockout animals has no factor A activity, and this is coagulation assay to look at clotting of the blood. And obviously, so with factor A knockout, which is in red, there's no clotting function in this knockout animals. So this animal model really mimics the human hemophilia A patient phenotype. So this is the model that we used to test the drug product function. So this is the efficacy study that we did using this factor A knockout mouse model. So various dose of the AAV product ranging from 10 to the 10th VG kg to 10 to the 12th VG kg were injected into the factor A mouse model by IV injection. And then the expression of factor A was measured by ELISA. So as you can see that the knockout animals has very low, below 5%. It's about 3% of expression, just base level expression. And then by injecting the AAV factor A product, there's a very high increase of factor A expression. And also by activity assay, it's a chromogenic activity assay. We also see that the activity of the product was as high as the wild type animal. So this really suggests that the AAV product was able to restore the function of factor A and to rescue the phenotype of hemophilia A knockout animal. And so we looked at also infectivity and look at viral copy numbers in the animal model. And so this is done by quantitative PCR. And this look at a viral genome copy number and over a time course. So this is months from 0.5 months till 7 months. So we look at both the AAV Cas9 copy number as well as the AAV factor A copy number. And we see a high expression of both the Cas9 and factor A virus expressed in the liver. And we also look at expression at the mRNA level. And this is just to measure mRNA level. We see a similar trend as the previous slide, which is the vector, the AAV vector copy number. And the mRNA coincides with the viral copy number. And again, at the protein level, so this is at the protein level as well as activity, also has the same trend as the mRNA level as well as the viral copy number. So all these data indicates that this particular AAV product was effective in treating hemophilia A animal. And this is to look at biodistribution of the AAV gene therapy product. And so we look at 2 time points, week 4 and week 12. And so these are the major tissues, and also we looked at the blood, and we were able to detect that AAV was present in these major tissues through this time course from week 4 to week 12, indicating that AAV expression basically was sustained for at least 3 months in the study window that we looked at. We also conducted a pilot toxicology study, and so this is just really to give us some sense about dose range. So like I said, the testing article is AAV factor 8, and we had 5 doses ranging from 10 to the 10th to 10 to the 11th VGkg. And we performed IV injection through the tail vein, and there were 6 groups of animals and 5 animals per group. And we're looking at, for safety, we're looking at clinical signs and liver enzymes. So because this is specifically to express in the liver, so liver is the main tissue for examination for toxicology here. And look at a PK/PD and immunogenicity. So that's another important part for AAV product is immunogenicity, either towards the AAV part or towards the therapeutic gene, the factor 8 part. And so there are different immunogenicity, including humoral and cellular immunogenicity. So there are also tests for immunogenicity tests. And at the end of the study, we also look at necropsy just to make sure, look at overall pathology of the animal after the drug administration. So this is just to show that looking at the liver toxicology, and because like I said, this hemophilia product is mainly to express factor 8 in the liver. So this is the most relevant tissue to look at. So so we looked at, basically we don't see any major liver pathology or toxicity. So these are the sections, liver sections, either vehicle, the control, or using the AAV product. And then we also looked at the liver enzymes, so albumin, ALT, and AST, these are liver enzyme, and we don't see a change between vehicle versus the product. So suggesting that there's no detectable liver toxicity using this product. So we also, for CMC, we also looked at the gene therapy product, its stability. So those are the things that we were looking at, sterility, genome titer, appearance, pH, infectivity, and potency. So for earlier studies, FDA allows infectivity to be a measurement for potency, but now they're really enforcing true potency assay to be developed before clinical trials can be conducted. So specific potency assays are very critical in terms of CMC. And then we also do purity assay to make sure the drug is pure. And these are performed at different time points, and 12, 24, and 36 weeks. And I just wanted to mention a few words of potency assay. So this becoming a very critical assay for gene and cell therapy. So basically, potency assay is really specific to each specific product, and it requires a robust assay, and that can be measured quantitatively. And so it's usually a cell line, it's a cell-based assay, and has to be product specific. and also has to be demonstrated over a range of MOI if it's gene therapy products. So it has to have a dose response curve and be reported relative to reference vector as a control. And so this is this potency assay also has to be eventually to be GMP compliant, so that it can be used as a law release specification for GMP. So that's why it's very critical in gene cell therapy development. So, so we did some potency assay for Hemophilia A gene therapy product. So this just gives one example to show that example of choosing what kind of cells. So I'm showing here the cell line that actually worked. So HepG2, because it's a liver, our drug is more liver specific. So HepG2 is a liver cell line, and we were able to show that using a reporter cell line, we were able to use HepG2 as a cell line for potency assay and GFP as a reporter, as a readout. And also the promoter makes difference. So you can either use a CMV promoter or another promoter, HCV in this case, and you see a difference between the two promoter. So the CMV seem to be a bit more robust in this particular cell line. So all these things need to be developed before you can define a specific specification for your therapeutic product. So I'm going to spend a few more minutes just to talk about the significance of using iPS cells for assay development. So this is really a new concept. So as you know, iPS cell has the potential to differentiate into any kind of lineage cells. So that's really the beauty of iPS cells. And this basically, iPS cells serve like a universal starting material that can be used for specific cell types and for specific product development. And another good thing is that iPS cells can be derived from patient. So then you can have patient-specific iPS cells. And if you don't have the patient-specific iPS cells, and gene editing can allow you to make specific mutations so that you can make those cells. So because of the combination of gene editing and iPS cells, we have the flexibility, we have the capability of making the iPS cells to be very suitable for assay development. Okay, so for example, in this case, we actually started with wild type cells and put in these point mutations. So this represents a familial protein variants of a particular protein. So these are just various SNP or point mutations in this one protein. These are just create different variants. And some variants are disease associated. And so the capability of gene editing in iPS cells really allow us to make more of isogenic cell line. And these are really good, not only for developing assays, but really for control, because you have isogenic cell lines to really compare the control with the disease cell line. And another thing I just want to touch on is our TARGATT technology. So our TARGATT technology is a gene insertion technology, and its really advantage over CRISPR is that it can do site-specific gene lock-in for large fragments. So I'm using here an example, and we're inserting 3 cDNAs, 3 genes, plus a reporter, mCherry, and this is more than 10 kb fragment. And we were able to insert it with TARGATT and at a very high efficiency. And for CRISPR, gene insertion lock-in, basically, the efficiency is relatively lower than TARGATT. So, for gene knocking, we usually use target technology to make these reporter or large fragment knocking cell lines. And so this is just an example that we have used target technology to investigate Parkinson's disease iPS cell differentiation by inserting the neuronal differentiation factors along with a reporter gene to trace cell differentiation. And another thing, as I mentioned, is that iPS cell differentiation. So iPS cells can be differentiated into all kinds of cells. So in this particular examples, iPS cells were differentiated into neural stem cells, into mature neurons, into astrocytes, and these cells, all differentiated from the one parental cell line, were subjected to 38 compounds and at two different doses, and that were used for neurotoxicity screen. So, that's a very good application of using iPS cell differentiated cells for assay development and screening. So last, I just want to throw in one example of using mouse models for the current pandemic COVID-19 study. So we have just generated COVID-19 mouse model. So we have generated two models. One is humanized ACE2 knock-in. So just going back a little bit. So really the cause of current pandemic is basically the virus enters into cells via binding to a receptor called ACE2. And so the mouse models is very desirable for testing. However, the virus does not bind to mice, the mouse version of ACE2. So, and previously there were models made. The human ACE was inserted into the mouse, however, by random insertion. And those mouse models do not develop severe disease, so they're not really usable. So our solution is to really make site-specific humanized ACE2 knock-in mouse model at genomic safe harbor locus. This guarantees that human ACE receptors are expressed, highly expressed in a safe genomic locus. And we also use a epithelial cell specific promoter K18, so that's more relevant because that's more in because ACE, the virus is more active in epithelial cells such as in the lungs. So that's why we want to express the ACE more in the relevant cells, that is epithelial cells. And then we're also generating a mouse model that has the mouse ACE gene knocked out. So it's a more pure humanized model without the mouse ACE model, ACE gene in it. So this really provides you basically using gene editing technology, using our target technology, we're able to generate such a model for potential development of cure for COVID-19. So with that, I'd like to thank you for your attention. And so please contact us at info@appliedstemcell.com for all your gene cell therapy preclinical needs. And we're here to support your gene cell therapy development. Thank you very much for your attention.